So I would like to introduce our two speakers for today. And so our first speaker we have with us is Megan DeWeerd. And Megan received an honors Bachelor of Medical Science in Biochemistry and Cancer Biology from Western University in 2019. She's currently a Master of Science candidate in Dr. Robert's, Dr. Robert's lab at Princess Margaret Cancer Centre. Her research focuses on high-grade serous ovarian cancer, and almost every patient with this disease has a mutation in the same gene. And Megan hopes to uncover cellular pathways that are reprogrammed by these mutations, which may lead to new therapeutic targets for ovarian cancer treatment. Next slide, please. And so along with Megan, we also have Julia Jaramillo. And Julia has received her Bachelor of Medical Sciences degree from Western University, where she specialized in biochemistry. She is also a holder of a Master of Science degree from the Department of Molecular Genetics at the University of Toronto. And she now works as research technologist at the Hospital for Sick Children, where she studies an adult brain cancer called glioblastoma. So I'm very excited to have these two presenters with us today, and we'll pass it over for the rest of the presentation.
Okay, hello everyone. I hope you're all doing well this, now afternoon. So my name is Julia, and thank you very much, Vinitha, for the introduction. We're really happy, Megan and I are here today to talk to you kind of a little bit about cancer research and a little bit more specifically about ovarian cancer research. So just to give you an idea, Megan and I are part of the Toronto Research Information Outreach Team, or RIOT for short, and we're affiliated with the Canadian Cancer Society. So this is a volunteer group. And we are essentially cancer researchers that like to communicate with the community as well as high schools to talk about a little bit about cancer research and the amazing things that are kind of going on in our fields. Now, we also post on blogs. We have a website called torontoriot.com that you can visit for blog posts that kind of cover exciting new topics in cancer research. We try to keep it pretty current, so you can check that out. Now again, we are cancer researchers and we volunteer with the Canadian Cancer Society. So our goal is really to share our knowledge with the greater community about current cancer research that's ongoing. Now, I just want to make a disclaimer, we are not licensed medical professionals. And so we're not qualified to give you any medical advice. So if you're searching for advice on a specific medical case, we advise you to consult your family doctor or oncologist for questions about that. Now today's outline includes essentially four main topics. So first, we will begin with an introduction to ovarian cancer, and then we'll talk a little bit about ovarian cancer risk factors and a little bit of research with that. And then we'll go into a bit of a research snapshot. So Megan's project that she's kind of really focused on, so you'll get to hear about that. And then finally, we will end with cancer stem cell section, which is kind of a ongoing research area, which is very fascinating. And so with that, I will give it over to Megan, and she'll start us off with the introduction to ovarian cancer.
Great, thanks, Julia. And thanks again, everyone, for being here and for having us. Next slide, Julia. So just wanted to start with an introduction to ovarian cancer in general. So on the right of your screen, you can see an image of the female reproductive system, of which the ovary is a part. And as the name ovarian cancer suggests, next, Julia, ovarian cancer is initiated within the ovary or within the tissues that are very closely surrounding it. Next, please. Globally, there are over 200,000 new cases of ovarian cancer diagnosed each year. So this is a large clinical problem. especially since, unfortunately, there is a very low response rate to therapy in many people with ovarian cancer. So even if there is a response, there can often be resistance that occurs later on in the disease. Next slide. There are some main symptoms of ovarian cancer, which I'll go over here, but unfortunately they are quite general and non-specific. So this means that they are common to many other health issues. So just because you're experiencing these symptoms, it doesn't necessarily make you think of ovarian cancer right away. So some of these symptoms include gastrointestinal dysfunctions, abdominal pain or bloating, fatigue, shortness of breath, urinary problems, or even no obvious symptoms at all. So this is a problem because obviously if a person is developing this disease but not experiencing any symptoms, it's likely to progress to a later stage before it's detected by health professionals. Next slide. So this diagram just goes over the stages of ovarian cancer. So in stage 1, the cancer begins within the ovaries, but it hasn't left the ovary at all. And this can either be in one or both of the ovaries. In stage 2, the cancer leaves the ovaries and spreads throughout the reproductive system. In stage 3, this spreads even further within the abdomen. And then in stage 4, finally, the ovarian cancer will spread to other areas of the body. Next, Julia. So stage 1 is obviously the ideal scenario where the cancer is still limited to one specific site. And then surgeons can go in and remove the ovary or both ovaries and take out all the cancerous tissue. And this leads to good cure rates. So this is what we want. But unfortunately, as I mentioned, spreading of ovarian cancer within the abdomen happens quite quickly, and it's not often detectable until later on. Next slide. There are several treatments that are available for people with ovarian cancer. So the current standard of care consists of surgery, if possible, to remove the ovaries and the fallopian tubes, which are attached to the ovaries, as well as any other organs that are displaying signs of cancer. So this could include the uterus, for example. People with ovarian cancer also often receive chemotherapy. So this is a type of medication that kills cells in the body that are dividing quickly. So that's essentially the definition of cancer, cells that are dividing uncontrollably. However, there are other cells within your body that do divide quickly in a normal scenario. So some examples include the cells that make up your hair follicles or the cells within your intestine. So unfortunately, chemotherapy can lead to some toxic side effects such as hair loss or nausea or other GI problems due to this kind of cross reactivity with some normal cells. Chemotherapy can also be used in combination with other drugs. So in this way, you can use lower doses of the individual drugs when they're paired together and hopefully improve outcomes for people with this disease. And finally, there are some hormone therapies that are in existence for ovarian cancer. So since the ovaries are receptive to estrogen and progesterone throughout the menstrual cycle, there are some therapies that can target the receptors for these different hormones. Next slide. So now that we've introduced ovarian cancer as a disease, I want to go through some of the risk factors that are in place for ovarian cancer. So starting with cancer in general, there are 3 main types of risk factors. So first is just random processes. So these we have no control over. As we age, obviously the cells within our body are dividing many more times than when we were younger. So a person who's in their 50s, for example, their cells have divided more times than someone like me in my 20s. So this we have no control over, it's just age dependent. Secondly, we have external factors. So these are things that we can control, such as exposure to UV light or smoking or any other kind of chemical or environmental carcinogen. Finally, we have heritable variants. So these are genetic changes that are present within you when you're born. So this could have been something that your parents had, or it could be something that happened within the egg or the sperm that eventually made up you. So these are the 3 different categories of risk factors within all types of cancers. Specifically within ovarian cancer, we can apply some different risk factors in these categories as well. Next slide, Julia. So for external factors, as I mentioned, the ovary is receptive to estrogen and progesterone. So any factors that change the levels of these hormones within your body could be a risk factor for this disease. So for example, in trans individuals who are receiving hormone replacement therapy or other reproductive factors. So if you choose not to have children or are unable to have children or have children later in life, because you're not exposed to those pregnancy and birth-induced hormone changes earlier in life, that can result in a mild increase in your risk. But again, as mentioned in the point below, this is a mild risk increase. So just because children aren't necessarily part of your life, that shouldn't be an immediate cause for worry for ovarian cancer. And as I mentioned previously, there are always those random events that are just due to age that we have no control over. Next, Julia. So there are some heritable factors for ovarian cancer. When we look at twin studies, it's been shown that the heritability of this disease is about 40%. However, we've only found about 15 to 20% of these genetic factors that increase risk. So within the field of ovarian cancer research, there's this large underestimation of these common heritable factors. So in a couple slides, I'll kind of get into where we can start to look for these missing factors. Next slide. So what are these risk factors that are inherited when we're born? The majority of these are mutations in proteins that help repair our DNA. So these are repair defects. So in this diagram here, we're showing one strand of your DNA, and your DNA is composed of 4 letters. Those are shown in these different colors here. And just like sentences and paragraphs, these letters are strung together in a specific sequence to specify a certain outcome. So in this case, the outcome is you. So your DNA dictates what your cells do and what they end up composing. So obviously this sequence is very important. However, there can be changes in the sequence that happen. These are called mutations or variants. So in this case, this T letter is changing to a C. But most of the time, there are proteins within your cell that can repair these accidental changes. So you can see in this diagram, the C has been changed back to a T. Next, Julia. So if you inherit a defect in this repair process, where you're missing a repair protein or it's changed, so it's not functional, these mutations or these accidental changes can persist within your cells. Next, Julia. And just one more. So these changes can accumulate over time. So now you see there are 3 mistakes within this small segment of DNA. And next, this can then increase your cancer risk. So if you're unable to repair these changes, you're more likely to have one occur in an essential protein that, when missing, can lead to cancer. Next slide. So as I mentioned, the majority of these inherited risk factors in ovarian cancer are responsible for decreasing your capacity to repair your DNA when it gets changed. And most of these factors are actually common between ovarian, breast, and prostate cancers. So any new research findings in breast cancer, for example, theoretically should be able to apply to ovarian and prostate and vice versa. So we can translate our research findings within these different cancer fields to each other. Next slide. So again, how do we find these factors that are missing, that we don't know exist, that are responsible for this inheritability of ovarian cancer? one thing we can do is look at past data from people with ovarian cancer. So we can look at the sequence of their DNA and compare it with other people with ovarian cancer and look for changes within their DNA sequence that might be responsible for this disease. So shown here in this diagram, we've got a person in green who has mild or indolent ovarian cancer, and then the red person has more aggressive disease. And we're comparing sequences of DNA. They're shown as those rectangles. And you can see the person with more mild disease has this large segment of DNA shown in blue that's missing in the person with more aggressive disease. So through these comparisons of past data from patients, we can compile a list of changes within the DNA within these populations of people and look for these changes that are common in a large population that might be responsible for this inheritance of ovarian cancer. Next. And we can use these new identified variants we can use them in comparison with other data from these patients to look at their risk probability and prediction of treatment. So on the bottom left, we can see 3 different variants that were identified within this population of people. And we're showing outcome over time. So as the slope goes down, the outcome is decreasing. So you can see that people with variant 1, they had their outcomes stay higher over time than people who had these other 2 variants. So identifying these changes in these population of people with ovarian cancer is really important for identifying them in future people who are diagnosed with ovarian cancer and kind of stratifying their risk at the beginning of their treatment. We can also use this data to predict for different patients or different people whether they'll benefit from certain treatments. So the graph on the bottom right here is showing 2 different treatments, treatment A and treatment B. And you can see that the outcome was better for people who received treatment A than people who received treatment B. So we can use this past data from people with ovarian cancer to inform future treatment of patients. Next slide. And again, this doesn't have to be necessarily with this past data. We can also do this with people who are newly diagnosed with ovarian cancer. So we can take a DNA sample, whether that's through blood or through saliva, and look at your sequence and conduct a risk assessment. So we're looking for those inherited changes, these variants that we've identified. And in this way, we may even be able to screen for earlier detection. So, for example, the Ontario government provides funding for assessment in high-risk individuals. So people who have a very extensive family history of ovarian, breast, and prostate cancer, you're likely to have one of those repair defects that are common across those 3 diseases. So that's funded to get screening for that. But there are also some other ways to get screened, such as through the Screen Project at Women's College Hospital, and there's a fee associated with that. But again, always consult with your doctor or your oncologist before looking into any of these avenues. We can also use your genetic information to kind of tailor your treatment plan towards the individual. So again, if you have these certain repair defects or other variants that are identified, we can then look at targeted therapies that are specific for those changes that might give you a better outcome. Next slide. So having gone over an introduction to ovarian cancer and its risk factors in general, I also wanted to provide you with an overview of my project. So as Misa mentioned, my project is focused on ovarian cancer, and I want to give you an idea of where that field of research is headed. So next slide. So my project focuses on a very specific subtype of ovarian cancer known as high-grade serous ovarian cancer. This is the most common type of ovarian cancer, accounting for approximately 75% of cases. So on the left here, you can see a diagram of the abdominal cavity, and the bottom 4 boxes there are showing 4 different common subtypes of ovarian cancer, and high-grade serous, the one I'm studying, is shown circled in red. And although I mentioned at the beginning of the presentation that ovarian cancer initiates in the ovary, in the past decade or so, research has shown that this specific subtype, high-grade serous ovarian cancer, actually starts in the fallopian tube where it attaches to the ovary. So although disease is often found within the ovary after it spreads, it actually starts in the fallopian tube. And as mentioned in my introduction, nearly every single patient with this subtype of ovarian cancer has a mutation within the same gene. So this affects one protein known as P53, which is involved in DNA repair. So again, it's somewhat of a repair issue. And it's also involved in regulating cell death. So if your cell accumulates those mutations, there are mechanisms within the cell, such as P53, which tell that cell to die so that it doesn't become cancer. So P53 is really important for repairing issues and also ending them if they persist. Next slide. So within high-grade serous ovarian cancer, these mutations in P53 are actually the very first step in cancer progression. So if you look on the image on the left here, we start with normal fallopian tube, which is composed of these pink and blue cells in this diagram. And because the end of the fallopian tube is so close to the ovary, it's exposed to the same environment as the ovary, and that's shown as these blue and green circles floating around in the diagram. So even though it's not exactly the ovary, it's exposed to the same things, and these factors can sometimes cause accidental genetic changes, such as a P53 mutation. So again, this is the first change that happens. And once it's mutated, you're more prone to accumulating more changes, as I mentioned before. So you progress to a high-risk scenario and eventually full-blown cancer, which can then spread to the ovary itself and also throughout the abdomen. So as I mentioned, P53 is important for DNA repair and regulating cell death. So logically, you would think that when it's mutated, when it's not normal, more mutations and more changes would accumulate and the cells would be unable to die. So this is logically what happens in many other cancers with mutated P53, where it's completely lost. However, next, P53 is not lost in the subtype of ovarian cancer, but it's actually stabilized. So this image on the bottom right is showing a stain within a fallopian tube tissue for the P53 protein. So in the first panel, in the normal fallopian tube, you don't actually see any stained P53 because in a normal scenario, it's constantly degraded unless it's needed. So if there's DNA damage, it will be activated, but otherwise it's never stable within the cell. Once it's mutated, in this case though, instead of losing it completely, it's actually stabilized. So this is the full protein just with 1 or 2 specific changes to it. So it's likely that it does still have some function. It's not actually lost. And you can see that these changes do persist throughout the whole progression of this disease in the high risk and the cancer state. Next slide. So my research is focused on this problem, and I have a few key research questions that I'm hoping to answer. So first of all, broadly, how are these P53 mutations leading to cancer? So this is the first initiating event, and clearly it's super important for the development of this disease. So what about this mutation is leading to cancer? And instead of losing its function, since it's stabilized, does it actually gain new abilities? So I'm performing a bunch of different experiments to look at new interactions with the DNA and new interactions with other proteins, and then the consequences of those changes. I should also mention that although every patient with a subtype has a mutation in P53, these mutations can be different. So it can be at different sites within the protein. It can be, a variety of different changes. So I have a couple of the most common mutations within people with ovarian cancer. And so I'm going to look at if these different changes, although they are within the same protein, they might have different consequences. And finally, can we exploit these new abilities or new vulnerabilities to create a new targeted therapy for high-grade serous ovarian cancer? So this is not an inherited variable, so it's not going to be used for earlier detection of this disease, but it could be a new therapeutic avenue if we can identify these changes and then target them with a new specific drug that will be more specific for this disease rather than the human body as a whole. So that is the goal of this project. I don't have any data to show you today, but hopefully this gives you a better idea of where the field is heading and how we're working towards creating new therapies for this disease. And now I just want to hand it over to Julia. If you guys do have any questions, please put them in the Q&A section at the bottom, and we'll get to them at the end of the presentation.
Thanks, Megan. Yeah, I just wanted to pause quickly and make sure that things are making sense and we're going at a good pace. So now's your chance to kind of speak up if you want us to slow down or if you have any issues. Okay, so we'll just continue then. Our last section then is about cancer stem cells. Now, I'm sure many of you probably heard of stem cells, but maybe not cancer stem cells. So cancer stem cells are kind of important for a lot of different types of cancers, and they're kind of becoming more appreciated. So we've heard previously that cellular diversity is really reflected by mutations, right? So we have genetic diversity, so lots of mutations that kind of work together within the tumor, and we have such different cells with different mutations that can make these cancers very difficult to treat. Now, one other component of cellular diversity is behavioral diversity. So how a cell behaves, how it functions, and essentially what's known as its phenotype. So a lot of the work with cancer stem cells has actually been with other types of cancers, such as leukemia. That's kind of the major cancer type where cancer stem cells have been studied. Now, we're going to speak a little bit broadly about cancer stem cells in general before we kind of get into specific ovarian cancer specifics. And so, moving on. So in order to really understand what cancer stem cells are, we just need to cover what like normal stem cells are. And so what is a stem cell? Well, before we kind of get into what a stem cell is, we can sort of imagine that, our bodies are made up of trillions of cells, and each with an important job that it carries out to make the body run effectively. So for example, we have these red blood cells, they carry oxygen all over your body. Brain cells, these neurons are sending signals to other neurons to tell your body to run or to breathe. So when these cells become specialized to carry out these specific functions, we say that they're differentiated. So that's just a key word to keep in mind. Now, the question is, how do we make all of these specialized cells? So the answer is actually that they come from stem cells. So stem cells are essentially undifferentiated cells, and they retain the flexibility to mature down different paths. So we have, for example, you may have heard of embryonic stem cells. So those can actually give rise to all different types of cells. Now, in adult tissues, we also have tissue stem cells, which are a little bit more specialized, but they still remain undifferentiated, and they can give rise to, for example, in the brain, nerve cells, or in the blood cells. So they play a really important role in maintaining the stem cell pool. So again, stem cells sitting at the top of the hierarchy, and they can give rise to these differentiated cells. Now, what are the properties of stem cells and why are they important? So firstly, dormancy, right? So the low rate, which is a low rate of cell division. So why is this important? Well, if we have these stem cells in our bodies that in the adult tissues can persist throughout life, we want to maintain these stem cells in the optimal state. So we want to prevent any kind of accumulation of mutations. And now with division, cell division, we always have a chance of acquiring mutations. And so if we restrict the division, and if we lower the rate of cell division, then we lower the risk of accumulating these mutations. So that's important because if you have a stem cell that acquires a mutation and then it gives rise to lots of different types of cells, all of those cells would end up with the same mutation. So that's why it's really important to maintain an unmutated stem cell pool. And secondly, endogenous protective mechanisms. So in stem cells, they actually have mechanisms that that kind of protect them from drugs or toxins, and it really preserves their integrity so that they can carry out their function over lifetime, so that they're maintained and so that they can continue to sort of regenerate your cells. So for example, leading to the third point, tissue regeneration and maintenance. So yeah, as an example, red blood cells are replaced every 3 months. So we need to make sure that we have these stem cells in our stem cell pool available so that when we do need these specialized cells, red blood cells, they can provide them. And so that's the critical part of maintaining the stem cell pool and maintaining them in a healthy, optimal state without mutations. And also the lining of your gut and your intestines can be replaced every 3 to 6 days. So we need to be really sure that we have that stem cell pool in order to be able to produce these highly specific or specialized cells. So now, how do these properties relate to cancer stem cells? So interestingly, these cancer stem cells are essentially a subpopulation of the tumor that have these stem cell properties. So when it comes to dormancy, this actually allows these cancer stem cells to resist traditional cancer therapies because those therapies mainly target highly proliferative or rapidly dividing cancer cells, right? So you can imagine radiation, chemotherapy can be very effective, but they are very effective on the cells that divide quickly. So if you have a subpopulation within the tumor and they're more slowly dividing, they will resist this treatment and they will be left over. And in terms of endogenous protective mechanisms against drugs and toxins, this actually confers drug resistance to these therapies, right? So they would be more efficient at either like pumping the drug out of the cell so that they're not affected, or they might have better DNA repair mechanisms. And finally, tissue regeneration and maintenance. So this is probably like the most fundamental part, is that these cells actually have the ability to reinitiate the cancer. So that is often seen or is believed to underlie the reason for relapse. So how does this really look? So this is kind of a diagram that illustrates these points. So we have our bulk tumor with the gray cells, the blue cells, and white cells all kind of mixed in, and then we have our yellow cancer stem cells in the center shown here. And when you add traditional cancer therapy, we have this drug that kills the tumor cells, but you can see that the yellow cancer stem cells have not died off. They persist, and that's because they are slowly cycling. And another term for that is quiescent. And so, although we've kind of debulked, we've removed most of the tumor, these cancer stem cells remain, and then they're able to actually reinitiate the tumor's growth. And so that's shown just in this slide here. So now, in terms of cancer stem cells in ovarian cancer, it's definitely an ongoing field of research, and it's a little bit controversial. It's controversial only because of certain markers. So people try to isolate these cancer stem cells with proteins that are specifically expressed on their surface. And this is kind of challenging because there's such heterogeneous cells that sometimes there might be more than one protein expressed on these cells. And so it's difficult to isolate these populations this way. So, but nevertheless, there's still evidence that there are subpopulations of cells within these tumors that have stem-like signatures in terms of the genes that are expressed, or that have drug resistance, kind of similar to stem cells, and that are able to initiate tumors. So now, one other aspect that is currently kind of a controversial topic in ovarian cancer research is actually the cell of origin. So now it's important to keep in mind that the cell of origin is not necessarily the cancer stem cell. The cell of origin is where the cancer began. And the cancer stem cell is the one with the properties that enable it to initiate and maintain tumor growth. Now, the cell of origin is still being investigated because we don't know if the cell of origin was a stem cell that lost its way and endowed the cancer cells with stem-like properties, or maybe the cell of origin was a more differentiated cell that reacquires these stem cell features and becomes undifferentiated. So there's likely different, it's likely actually a different cell of origin for different subclassifications of the ovarian cancer. But why does this really matter, right? Well, maybe it doesn't, or maybe by identifying the cell of origin, we can have a better understanding of how a normal process went awry. And that could help with prevention, early detection, and treatment. So these are just some topics in cancer and ovarian cancer research. But going back to the cancer stem cell population, so how can we target these cancer stem cells? We know that they're believed to really be responsible for relapse. And so what's one way of targeting them? So first, we can actually target them with a specific drug that kind of specifically targets these cells and kills these tumor stem cells. And this would allow the tumor to then lose the ability to generate new cells, and then ultimately the tumor would shrink. And we wouldn't be able to get that regrowth because we've eliminated the cancer stem cell population. Now, another way to target these cancer stem cells is to actually make them more sensitive to traditional cancer therapies. And so, for example, we can use compounds that actually push them to divide more quickly, or push them out of quiescence so that they will be killed by the therapies that target rapidly dividing cells. So that's how we would essentially sensitize them to the traditional cancer therapy by eliminating their stem cell properties, such as their ability to remain quiescent. Now, I'm just going to give you a couple of examples of how this actually worked. So for the first study, the title was Targeting Notch, a Key Pathway for Ovarian Cancer Stem Cells Sensitizes Tumors to Platinum Therapy. So just to give you a quick background, Notch signaling is really important for maintaining stem cells. And then the diagram on the right, it kind of just is a simplified version of how it works. So we have two neighboring cells, one with the Notch activator and then one with the protein that then sends the signal to the cell. And so once it's activated, it then goes to the nucleus and then drives the production of these stem cell genes. Now, interestingly, Notch signaling has been found to be active in ovarian cancer stem cells. So inhibiting Notch signaling is one way to target these ovarian cancer stem cells. So we know that it's a stem cell maintenance pathway, and so if we can inhibit this pathway, maybe we could eliminate the stem cell properties of these cells and make them more sensitive to therapy. And so that's actually what they did here in this paper. They showed that treatment with cisplatin increased the expression of cancer stem cell genes. So that kind of suggests that when you treat with this chemotherapy agent, you get maybe an enrichment of cancer stem cells, right? Because you get increased expression of these cancer stem cell genes. But if you then, and that makes sense, right? Because we're eliminating the bulk of the tumor cells, but then the ones that remain are the ones that are not dividing as quickly. So we're enriching for that population. But when you treat with this inhibitor of Notch signaling, you actually see decreased gene expression of stem cell genes. And so that's very promising, because then maybe you're eliminating that population of cells. Now, they showed that a combination of these two compounds resulted in the lowest cell viability in vitro. And that's shown here, where relative to control, you can see in the gray bar, it's much lower when the cell viability is much lower when you use the combination. And then finally, in preclinical models, they did find that this combination therapy resulted in better outcomes. So this is one way to demonstrate how you can target, the bulk tumor cells, but then also the cancer stem cell population. Now, for our study number 2 that we just want to cover quickly, it's titled Autophagy Inhibition Reduces Chemoresistance and Tumorigenic Potential of Human Ovarian Cancer Stem Cells. Now, what is autophagy? Well, it's the process by which cells can essentially eat themselves in order to maintain a balance. So early in cancer progression, it protects the cancer cell from self-damaging organelles like pro-oxidative mitochondria, and then later on, it actually prevents chemotherapy-induced toxicity and helps to keep the cell from starving by eating the things that it doesn't need. And so it's been found that in ovarian cancer stem cells, they actually have higher levels of autophagy. And so we can maybe exploit this by inhibiting the autophagy process using chloroquine. And that's what they did here in this paper. And they found that by blocking this process, which seems to be like what the cancer stem cells are relying on, they found that it reduced characteristics of cancer stem cells. So in this figure here, it's just showing that relative to the control, you can see that the cells are making much smaller spheres, which is the ability to form spheres is a property of these cancer stem cells. And they also found, in addition to this, that there was increased sensitivity of chemotherapy in vitro, and that blocking autophagy and giving chemo reduced tumor growth in mice. Now, further work is needed, but it definitely is an interesting paper and shows some promise. And with that, we'd just like to thank you all for listening to our talk, and we hope that you learned a lot, and we'll be taking some questions